Okay.
So everyone got food yesterday.
Chick-fil-A? You know, I did Claire has a new sandwich with turkey. It's like an egg, some great tomato pesto, roasted turkey. It's really good. Anyway, normally I don't think about it for breakfast. Okay, guys. Okay, are we ready? Okay. So let's start where we left off. Okay. Gene expression topics clearly should be finalized. What I meant to write is just that your aims need to be finalized this week. So that should be your goal, to have 3 aims by the end of this week. Most people that I've seen so far, I guess that was on Monday, are pretty much there. There's been a little bit of help that's been needed, but that's to be expected because this is the first time a lot of you guys are doing this. There's also been a fair amount of frustration among people who are having a hard time understanding the project. That's also normal because we all get frustrated when things don't come easy to us, right? So just power through and it'll be okay. Exam 3 is next week. That reminds me, those of you who are interested in an oral exam, please email me. I've already had one person actually who's emailed me about oral exam for this time. It is a possibility. Okay, so if you're failing and you don't know why, if you feel like you understand everything, but you're still not doing well in the exams, an oral exam might help boost your grade, or it might not. If you really don't know something, you don't know something. I was feeling like I was talking and listening to other conversations at the same time. It was so weird. Okay. Multiple choice questions. On average, people have been submitting less and less questions each time. Well, I guess now it's only the 3rd time. But usually at least one gets in. So those of you who would like to try your luck at submitting a multiple choice exam question, please do so by end of day Friday. Okay? What else do I have to say? That's it. Semester is coming to an end. It's really quickly, isn't it? Good thing. Okay. I'm getting tired. It's hard. 408 and then 409, right? Who did 408 and 409? Who feels tired after that? Yeah, that's a lot. Okay? Okay. So, this is what we've done so far. This is the same list we had the other day, and that's because we're still talking about regulation of gene expression. And today we're going to try to tie up the rest of activators and repressors, and then get to some good examples of how gene expression is regulated in eukaryotic cells on Friday. Let's see if we can do it. Okay, so this is where we ended. There's so much talking today. Do I look funny? Am I wearing, I mean, the colors, actually, I had a quandary. Because this color blue and this color blue are funny, and then the blue shoes, different color, but actually having multiple shades of a color is okay now. Just as multiple patterns, right? Animal print and stripes, no problem. Who reads Lucky Magazine? Okay, fine. Anyways, I just want to get, everyone's here. It's going to be a beautiful day, but it's not warm yet, so no spring fever allowed until afternoon. Okay, so we started talking about activators and the concept that they're modular, meaning they have an activation domain and a DNA binding domain. And I introduced this at the end of class. Whoa, there, wrong button. Sorry. Okay, so here's how it looks, right? So you've got your activator binding to some DNA element, and it has a DNA binding domain that binds to DNA, and it has an activation domain that's going to interact with something going on down at this end, because remember the DNA kind of folds over and leaks out, and there's a mediator and all these proteins involved. So people have actually used this to study just how activators work, and then now developed whole research tools based on this concept. So to think about the concept, you can think about separating your activation domain from your DNA binding domain, right? So if you have your DNA binding domain and your DNA sequence here, you can actually activate whatever gene you want by switching out your activation domains, okay? So you engineer your whatever to have some plasmid or whatever to have a certain recognition sequence, and then you put in your oh, wait a minute, I'm confusing something. So you put in, sorry guys, I was thinking about something else. So as long as you have the ability of your DNA binding domain to recognize the DNA, you can activate anything, right? So here you've got LEX-A, which is a different gene, okay? So up here you're activating gene expression for galactokinase, and here it's LAC-Z. And so if you have a chimeric protein that has the DNA binding domain for LEX-A, but it doesn't recognize the LEX-A sequence here, you're not going to have gene expression because it needs to bind to the DNA to activate expression, even though you have the activation domain, right? Because if it can't bind, then it's floating out in space and can't activate gene expression. But if you have the recognition sequence for LEX-A, then the DNA binding domain can bind and activate gene expression of a different gene. Okay? So let's talk a minute about how this is used in biotechnology or just research in general. And I'm going to introduce the concept of the yeast-2 hybrid. Who's heard of a yeast-2 hybrid? Oh, great. Who's done a yeast-2 hybrid? Any either of them? Actually, I've been in many labs that have, and it's very cool. So it's a way to study protein-protein interactions. It's a way to go fishing. Let's say you find a protein and you want to know what it interacts with, and you've got no idea. One way to do it is to do a yeast-2 hybrid. Okay? So you're using this concept of modular transcriptional activators to study protein-protein interactions. You have a DNA binding domain and an activation domain. That's what DB and AD are standing for here. And you actually separate those two components of your transcriptional activator and fuse them to proteins. Okay? And so what you're doing is you're looking to see if the proteins will interact, and that will bring both domains together to make a functional transcriptional activator. So what you're doing is you've got protein X and protein Y, and you've engineered them. So you've actually pasted together the DNA sequences for your DNA binding domain and your protein of interest, and your activation domain and your protein of interest, and you put them together in yeast, and if they interact, then they form one complex, right? So they come together and can activate gene expression. Pretty cool, right? So how would you use this? Why would you use this? Well, if you have, so here's another way to look at it, right? So if you've got your DNA binding domain in the same cell as your activation domain, and your proteins interact, they come together and form a functional activator. If they don't interact, they're not going to come together. And so then what you do is you plate this is all happening in yeast, and you plate on some kind of selective media, and you look for gene expression. So remember, you're activating gene expression, and so you're looking for a reporter, activity of some kind of reporter. Let's say it's something that's going to turn your cells blue. Or let's say it's something that's going to either allow growth or not. There's multiple ways to do it. But here, you're looking for blue because you're getting expression of a gene that's going to turn your cells blue. Okay? So when you have interaction, you're going to get expression, and when you don't, you don't. And then you can actually look. You know what you're plating, and then you can look and see, oh, when these two proteins interact, I get growth. That means they interact. And you can start to identify what proteins interact. So you're saying, okay, what does this have to do with anything? So let's think about how you might use this kind of assay and relate it to the CMV project. Okay? So you're studying CMV and its effect on the immune response. Have any of you guys done that before? I hope everyone in this class says yes, because that's what you're doing in your project. Right? Everyone can relate to this because this is what you guys are doing right now. The number of people who are like this, you guys are all going to get a beating later. Okay. So it appears that CMV down-regulates gene X. You guys have all found your gene X that's either up or down that you're interested in, right? And you're trying to study the mechanism. So if you're really doing research, you would be trying to study the mechanism, right? So you suspect that some CMV gene, we'll call it gene A, that it's responsible for this downregulation, but you have no idea what it might be doing. And so you decide to use a yeast-2 hybrid assay to see what that gene product, which is a protein, might interact with in the human genome. Okay? So you've got your bait. You've got your bait, which is your DNA binding domain, right? So it's got CMV gene A on it. And then you've got your prey, which has your activation domain fused to it, and then that's a whole cDNA library. So every gene in the genome, separately, not all together. And so these are all on plasmids in yeast. So you can actually go and buy yeast where you've got a yeast cell clone that contains every single gene in the whole genome. You can do that. Go buy it. It's online. It's expensive. Not that expensive. So then you can mate your yeast. So you want to end up with the plasmids in the same yeast, right? And then you grow them on selective media. Why selective media? Because you don't want to grow something that doesn't have plasmid in it, right? And you look for your color change resulting from expression of your reporter gene. So you do this, and you find these blue colonies. What do those colonies represent? Who wants to raise their hand? I can call random people. Yes. Activation of gene expression of your reporter. But what does it represent? Yes. Gene A, or protein A, because really it's going to be the protein, is interacting with the other protein, with some other protein out of the genome. Okay? And so why is this useful? Why is this useful to study the effect of CMV on the immune response? See, someone who hasn't spoken yet. Right? So if you're very interested in the effect of CMV on the immune response, and you think that some CMV protein is interacting with some human protein, how is that useful? Yeah.
Probably use it to figure out a way to deactivate that CMV protein.
Long term, you can look in terms of, well, let's deactivate or intervene in some way. But what does it directly mean? What does it mean about two proteins? If you see two proteins interacting, what does that mean? They're probably working together somehow, right? If proteins bind to each other, they're probably doing something together. And if you know CMV is downregulating something, and then you see an associated protein that you pull out of the yeast-2 hybrid interacting with that some CMV gene that you think is involved, it leads you down a path to understanding the mechanism by which CMV is affecting the immune response, or whatever gene that you decide to focus on. Okay? Who thinks I'm talking in some weird language that's not English? Who thinks I'm talking in Pig Latin? I can't say Chinese because some people might speak Chinese. So, Pig Latin? No. Who used to speak to their friends in gibberish in like elementary school? Okay. Anyways, I had some friends and we did that. I don't know. It was like 4 years of us just talking. And anyways. Okay. Who's like, what is she talking about? Someone raise your hand so you guys all get it. Now I want you all to take a moment to think in your mind about the project that you're focusing on for your gene expression project and say, would a used-to hybrid be useful? Just take a minute. If you're sitting near someone in your group, you can talk to them, or you can just think about it. But just take a minute. Is this useful? How could you apply this method to studying whatever you're doing?
Yeah. The interaction is transient, like, kinase comes to phosphorylate it, it's not sticking out there. Is this going to show up those type of players that have this transient interaction?
So I just, well, oh wait. Like now, right? Phosphorylation is also be slightly different. Right? So I'll here's my
but this is going to show us, like, because if they are involved, they're likely going to be using it the same way to do that data.
Great. Start long enough, like if you're doing an activation domain together long enough, well,
So if GPs versus identified as
okay, guys, I just got a few questions. Who thought about this and said, you know what, this makes, I can see how I might apply this? Who has no idea, very good, who has no idea how they might, okay, so you guys are having a hard time figuring out how to apply it. Okay, so bring it up in recitation with me if you think it could be useful and you can't figure out how to apply it. Now I'm not saying, I do not want everyone in this class to come with a U2 hybrid in their proposal. But it's useful to be able to apply concepts, because then you understand them. So I got a few interesting questions. One I got is, can a U2 hybrid detect transient interactions? Because don't forget, every interaction in the cell is not like, boom, we're binding super, super tight forever, right? You might have this brief binding, but as long as you have your DNA binding domain and your activation domain close enough to form a functional activator for a long enough time to activate gene expression, it's going to detect that interaction. The other question is, what if the interaction requires some kind of post-translational modification? You might not see it, and so there are limitations to a U2 hybrid. It's not the be-all and end-all. Many years ago, like 10, 15 years ago, people thought it was the be-all and end-all to studying interactions. Now there are a lot of different ways to study. Yes?
Can it help you see, like, what kind of domains they have the protein?
Can it help you to see what kind of domains proteins have? Well, it's detecting whether two proteins can interact. Let's say gene A, CMV gene A interacts with, you get 50 hits. Sometimes you'll get like 50 hits, you're not going to get one. And you look and see what all those proteins are. Maybe they'll all share domains, and then you'll be able to say, this CMV gene likes to interact with proteins that contain helicase domain, or whatever it is. And that could give you a hint as to what something might be doing. Okay. Any other thoughts? Yes?
So if you have your one gene fixed and you do this on all your whole library, like you're just looking for like a common fix for the gene who have like one line, or like how would you apply to your gene?
Okay, so what's your gene? IRF5. So you say CMV is doing something to IRF5. We don't know what, and like through future research you decided CMV gene X that's doing it, or gene A, whatever gene is doing it. And so then you put your bait and your prey in, and you get a whole bunch of hits of what that CMV gene is interacting with. And so then you'll go through them and you'll say which one is associated with the IRF signaling pathway. And then you'll be like, oh, well, CRB gene X interacts with this kinase that's involved in activation of IRF5 or FC or whatever. Maybe it's turning that kinase off, and that's what's resulting in downregulation. You understand what I'm saying? So it's really about this huge, you have this huge puzzle that you're trying to put together. And this is going to give you one more little bit of information. Okay, and it's a way to screen a whole genome. Okay, so let's move on, because now you're saying, what does this have to do with gene expression? It's just the use of we've created a tool from what we know about the modular properties of gene activators, okay? Okay, so eukaryotic gene activators increase the rate of transcription initiation. That's a good way to think about them. They attract, position, and modify the general transcription factors in RNA polymerase II at the promoter so transcription can begin. They act on transcriptional machinery and modify chromatin structure around the promoter. Okay? That's what they do. Now we're going to talk, now we're going to look at pictures. Okay. So, here's your activator. Here's your activator binding up here. You can see what it's doing is it's helping these general transcription factors, RNA polymerase mediator, this whole complex to assemble into an active transcriptional unit and activate initiation, okay? Without, you can imagine without this, or if you got rid of your activation domain, it wouldn't bind here, and so that then you would not be able to have this stable enzyme complex, okay? Yes?
Now, the mediator itself, does it change based on the activation domain?
Does the mediator change based on the activation domain? Mediator is a complex of proteins, and I believe the complex is fairly conserved in its function, but I'm not sure. I believe that activation domains, probably their domains, which means that they are conserved, and so you have a conserved unit interacting with a conserved unit. Okay? And really what's regulating this activity is can it bind to the DNA? That's your specificity, your DNA binding domain binding to a specific DNA sequence. Does that make sense? Okay? Confusing look on face? Would you like me to expand? Okay. Okay, so you can imagine on an exam I might say, deletion of the DNA binding domain on an activator is likely to do what? And you would have to evaluate what could happen and what couldn't, right? Okay, so an activator might also help with assembly of general transcription factors, right? So one is just kind of creating this whole complex to activate transcription, and sometimes it actually might help bring in certain transcription factors, general transcription factors, because remember, all of these need to assemble to bring in RNA polymerase. So that's kind of an extension of the last slide. They can alter chromatin structure, right? See, this is where you really need to start bringing in all of this information we've learned over the semester and integrating it together. So you can have, here's your chromatin and your gene activator protein binding right there. And your TATA box is there, right? All wrapped up in histones, okay? And so a lot of things need to happen for gene expression to happen. And so you might have your gene activator protein might recruit a chromatin remodeling complex and remodel nucleosomes, exposing the promoter. Histone chaperones might be recruited to remove histones. Histone chaperones might replace histones in different places. It might also recruit histone modifying enzymes, right, to modify histones to a conformation that's going to promote transcription, and all of those things need to happen so that general transcription factors, mediator, and RNA polymerase can assemble and activate transcription. Okay? I'll let that sink in. So, does anyone have any questions about this general concept? Are you guys all making connections between the first unit and now? Okay, because you should be. If you're not, you need to see me. Because this is major convergence of topics. Okay, so activators can recruit histone acetyltransferases that acetylate histones, recruiting more activators, right? So here's your activator, and it's binding a HAT, which is acetylating histones, and then different activators will bind to acetylated histone, okay? So another thing to think about is that TF2D, a subunit of TF2D, remember that's your first general transcription factor that binds, right, to your TATA box, or to your promoter, I guess you could say. It contains 2 bromodomains. What do you think what's that about? Right, so what does it tell you when I say there are 2 bromodomains on TF2D? What might it bind to? I'm hearing people talk. Go down the line. Where's Doug? A what?
The hats.
Right, so hats, what are hats doing? Okay, so hats acetylated lysines, TF2D binds. Remember, acetylation is involved in what? What's acetylation involved in? Activation of gene expression, opening of the chromatin, and right? So it kind of makes sense that TF2D might bind to acetylated histones. So let's see, what does this look like? Okay, so the histone code is very much involved in transcription initiation, right? So you've got your histone with your DNA wrapped around it, and there's your promoter and some kind of, here, this space here means there's a long distance, right? Then you have some kind of enhancer region. And so a gene activator protein might come and bind to that enhancer region and recruit a histone acetyltransferase that then might acetylate the histone tails, right? And then that might result in recruitment of some kind of histone kinase, which might phosphorylate histone tails. Remember, that also happens. We didn't spend a lot of time talking about phosphorylation, but it also happens on histone tails and creates binding sites for things, right? And then that might result in recruitment of chromatin remodeling complexes, right? And then that might result in recruitment of, then you might get recognition of acetylated, phosphorylated histone tails by TF2D, and then you get that cascade of general transcription factors binding to your promoter, and then transcription. So look at it a little more closely, right? So you'll have an activator binding to some kind of DNA element upstream, and then you get recruitment of a HAT. Now this is not every single time, but this is a likely thing that could happen before transcription, right? And you get acetylation, right? And then you get recruitment of other proteins because it's able to recognize acetylated histones, and then you get recruitment of kinases that are going to further modify histones. And then you get this histone code is being generated, right? Histone code is different modifications on histone tails that mean something, right? So you're starting to get lots of modifications, lots of acetylation and phosphorylation, which is resulting in recruitment of chromatin remodeling complexes, and then TF2D. And then you get TF2B and all these TFs, right? And then you get recruitment of your RNA polymerase, okay? So that's how initiation is regulated by activators affecting the histone code. modifying histone code, creating a histone code that means something, which recruits all the things that need to happen so transcription can be turned on. Okay? Any thoughts? Anyone want to share any stories with me? No? No stories? Okay. Okay, so what needs to happen for successful transcription of a eukaryotic gene? Whole bunch of things, right? You need an activator to bind to chromatin. You need chromatin remodeling complexes to remodel the chromatin. You need histone modification enzymes to modify histones. You need other activator proteins to come in to bring in additional regulatory proteins, to bring in mediator and general transcription factors and RNA polymerase. And then you have this pre-initiation complex at the promoter that can be activated. And that's how you get transcription initiation. Yes?
What's the difference in function between the chromatin remodeling complexes and the histone modifying enzymes?
What's the difference in function between chromatin remodeling complexes and histone modifying enzymes? Great question. Chromatin remodeling complexes are really moving histones around on the chromatin, on the DNA. So they're taking them off, putting them back on elsewhere so that you can access the promoter. Chromatin modifying enzymes are modifying histone tails or DNA through acetylation or methylation or whatever. So that's the HACs and the HDACs and the methyltransferases and all of those. Good question. So you can see how they both would fit in with initiation. Yes? Would histone modification enzymes come before chromatin remodeling complexes? Well, it just depends on what's happening. I don't know if you can put them in sequential order. It's really about what's recruited, right? So if an activator is going to recruit a chromatin remodeling complex, that might happen first. If an activator needs to recruit a histone modifying enzyme to modify histone to then recruit a chromatin remodeling complex, that can happen too. I don't think it needs to be one then the other. Yep. Okay. Any other thoughts? Sometimes it's good just to like sit and let it sink in for a minute. Sink in. Think about it. Okay. Okay. So I put this together so you can think about things in a very basic way. When asking about gene regulatory proteins and what they do, ask yourself, does this promote gene expression? If yes, it's most likely an activator, and activators can do anything that promotes gene expression, from recruiting HACs to recruiting general transcription factors. Anything that's going to promote gene expression. Ask yourself, does this promote gene expression, right? You know a HAC is going to promote gene expression. You know that an HDAC is likely not to promote gene expression. You know that a methyltransferase, well, it depends. So if your answer is no, ask yourself, is this actively inhibiting gene expression, right? If so, it's likely a repressor. And a repressor can do anything that it's inhibiting gene expression, from recruiting HDACs and methyltransferases to blocking assembly of RNA polymerase and general transcription factors. So you can think of it in two very clear groups, okay? So that way you don't feel like you're having to like memorize all these things. Okay, think about function and effect on gene expression. Okay? Okay, so let's talk about repressors for a minute. Just like activators can work in many ways, right? They could recruit many things that promote gene expression. Repressors can work in many ways by recruiting many things or that either promote repression or inhibit activation. Okay? So a repressor can bind and actually cover up the binding site for an activator. So it can competitively exclude binding of an activator. And so that's what this is showing. Right? So red is stop, green is go. Yeah. So repressor is binding, and it kind of shares a binding site a little bit and prevents binding of the activator. So that's one thing that could be That's one function of a repressor. It can mask the activation domain, right? So if you've got an activator and a repressor binding site right next to each other, they might, the repressor might choose to bind to the activator and prevent the activator's activation domain from interacting with anything else. Yes?
On the last one, if the activator got there first, was the repressor like outweigh it and kick it off?
So if an activator got there first, could a repressor pick it off? I don't know. I'm thinking it really is a numbers game. So when you think about proteins and what they do, you have to think about the expression levels of each protein. If you're trying to repress something, gene expression is going to result in high levels of the repressor and probably lower levels of an activator. So just by chance alone, you're more likely to get lots of repressors binding to the repressor binding site. That's the way I think of it. Does anyone want to add to that thought? No.
There could be a different affinity for the same region.
There could be a difference in affinity for the same region. So perhaps the repressor binds much more strongly to the DNA than the activator. But then again, you need to have a lot more activator then to kind of compete it out. So think of it as competition between binding. Yeah. Anyone want to add to that? Okay. Masking the activation surface. Also, could prevent assembly of general transcription factors and recruitment of RNA polymerase. So if you've got a binding site for a repressor and a binding site for an activator, that activator is going to normally recruit all these general transcription factors, a mediator, and RNA polymerase. But when the repressor is present, it might bind and prevent the activity of the activator. It might also recruit chromatin remodeling complexes that decrease the accessibility of the promoter. So the opposite of how an activator might activate transcription, right? So if you recruit a chromatin remodeling complex and close up your chromatin to obscure the promoter from any transcription factors, you're going to be repressing gene expression. It could recruit histone deacetylases, right? Right, so if you have a repressor here and it recruits a histone deacetylase, it's going to remove all the acetylation, right? And that's going to be associated with repression. It could recruit histone methyltransferases, so then you can methylate all your histone. So it kind of, you can see the direction we're going in is these are all the things you've already learned to associate with repression. And so now you're kind of understanding how that happens. Okay? So if it recruits factors associated with repression or turning off gene expression, it's likely a repressor. Okay? I guess I kind of flew through that. Who wants to ask questions about all that? Or does it just make so much sense that you're wondering why I even had to tell you? Good. No one's crying. Okay. Okay, so, wow. This is towards the end of my lecture, which means we did really well today. So eukaryotic genes are regulated by a committee of proteins. So many proteins are involved in regulation. And the transcriptional outcome depends more on the DNA sequence bound than the regulatory proteins themselves. Okay? So there are all these proteins. In solution, they might be able to interact with all sorts of things. But it's how they bind to the DNA that's going to determine what's going to happen. Right? So in some cases, you might get recruitment of proteins that recruit a coactivator that's going to activate transcription. And this same protein, in combination with different proteins, might recruit a co-repressor and repress transcription. So it's really about what's binding and what's being recruited and how that's affecting gene expression. So that's why I can't say, this one protein does this. So think of it in categories of repressors and activators and effects on gene expression. Okay. And so sometimes you'll have these huge number of gene regulatory proteins that are required to activate transcription. Okay? So just understand that it's not just one activator binds to one thing and then you get gene expression. There's a ton of proteins involved in activating or repressing gene expression, and that's how gene expression is so tightly regulated. Like in the CMV project that you guys are doing now, you can see that so many different pathways are being activated, right? And they're all involved in the immune response. But like NF-kappa B, people keep bringing up NF-kappa B. It activates like a million things. Why doesn't it all just activate one thing? But if you look at that whole gene list, you'll see some things that are activated by NF-kappa B aren't turned on by CMV. So it's because there are so many different factors involved in determining, and so many different binding sites involved in determining which genes will be activated under which conditions. And that's why there's so many proteins involved here. Okay? We made it through something like almost 30 slides. Wow. So that was kind of fast, and I'm sorry. I think I had too much coffee today. Does anyone have any questions about all that stuff? Or are we good? Because then you can go early, and then we'll just talk about examples. Yes, let's wait a minute.
That's not a question,
Oh, it's not. It's not.
But, I can't find my laptop. I left it. So I was wondering if
Oh, I didn't see anything. Did you check the bio office? Because there's been so many classes in there. That's scary. Sorry.
It's like someone can set up.